The final piece of new material for our year in AP Biology involves the second communication system of the body, the nervous system. And so, with this, we are going to again sort of provide a little bit more detailed examples of some of the things we've learned this year, ranging from cell signaling to just cell parts, and into things like exchange of materials across the membrane. We're going to look a little bit at the structure and function of your nervous system, very briefly at the brain, and then primarily just focused on how the nervous system communicates through your body. And of course, the baby memes continue. So, as we had talked about kind of previously, the other side of the communication aspect in your body is the nervous system, which is a system of highly specialized cells. These are some of the most specialized cells that you see in the animal body. And these are neurons. And what neurons are able to do is transfer information electrically. Now that sounds weird. It's not that your body is firing at some big high voltage or anything like that, but your cells can send these long distance signals basically using differences in charge. And then, as we saw previously, they will finish that with a synaptic signal, or a very short distance chemical signal. Now, if we start with just sort of some background and structure of the nervous system, neurons are what we call these nervous cells. Neurons go together to form a nerve, so you have a bundle that is a nerve running from a certain place to another place in your body. And then you have two very large bundles of nerves or neurons, which are your brain and your spinal cord. Now, the nervous system also includes some supportive cells called glia or glial cells. These do not communicate or transfer information, but rather these sort of support the neurons and help them to do their job. But maybe the most important thing of this first slide is in this little box here. Basically, the nervous system involves kind of three pieces, just like cell signaling involved three pieces. And that is sensory input, integration or making sense of that input, and then output, which is typically motor. But just another way to put it, just input, process that input, and then output. That's what your nervous system can do. Now, the nervous system itself is broken down into two large groupings or components, the central nervous system and the peripheral nervous system. The central nervous system, or CNS, is rather simple. It is just your brain and spinal cord, as you can see in this picture. The peripheral nervous system is a little bit more complex in its breakdown. The peripheral nervous system, or PNS, is where the input and output takes place. So where the central nervous system was kind of like your processor or your integration, the peripheral nervous system, all those other neurons and nerves, are your inputs and outputs. So technically we refer to inputs as the afferent neurons, and so these are your sensory neurons that allow you to sense the environment around you. And then efferent neurons are your outputs. And these outputs can kind of come in two forms as well. Outputs can be motor outputs, so signals that would help you to move, or outputs can be autonomic, which I just put here in general as signals to everything else. Notice this is not automatic, it's autonomic, but these are also kind of things that often happen without your conscious control. Now, the autonomic nervous system actually breaks down even further into three different groups. The first grouping in that autonomic nervous system are the sympathetic nervous responses. Sympathetic nervous system is like your fight or flight that we mentioned before, that response to stress. So the epinephrine we talked about in the endocrine system is kind of tied into this sympathetic part of your nervous system. Parasympathetic is the opposite. So parasympathetic, a lot of times we think of as your calming response. So sympathetic is the stress response. Parasympathetic is the calming response. And the last thing here we won't even mention again after this slide, but your enteric nervous system is your digestive system innervation. So this is obviously something that is completely, unconscious. It's not something you have to think about or you have any control over, but you do have some nervous control of the smooth muscle that helps move your food and helps you digest it. And this cracks me up because this looks so much like my dog Samson. Now here's a breakdown of the sympathetic versus the parasympathetic part of your nervous system. Very important here. Don't memorize this, right? This is just here for perspective. But you'll notice here on the right are some of those things that we had mentioned in the last set of notes that are part of that fight or flight response, right? And these are all things that your sympathetic nervous system is responsible for helping with. So it dilates your eyes, again, so more light comes in. You can see better if you would need to fight or flight. Actually inhibits saliva secretion, relaxes your lungs so that it's easier to bring in oxygen. Accelerates your heart rate, which will help get the blood and therefore that oxygen and that sugar out to the tissues that might need it. We're going to inhibit the stomach and intestines, as we talked about. You don't really need to digest food. Inhibit the pancreas for the same reason. Stimulates glucose release from the liver. That was really that job that epinephrine helped with a lot. Also stimulates the adrenal medulla, so this will lead to release of more epinephrine. Inhibits the emptying of the bladder, again, not something you need to do in a very stressful situation. And then on the left-hand side, you see those parasympathetic things, which again are sort of just your opposite things, right? Pupil of the eye constricts, stimulates salivary secretion, constricts the lungs, slows your heart rate, stimulates activity of the stomach and pancreas and all that good stuff. And notice that most of these parasympathetic tracts come from right up here by the brain, whereas those sympathetic tracts occur kind of along the spinal cord. But again, do not spend time studying this chart or anything like that. Now again, neurons can have three basic functions which go with those three basic steps that I mentioned on a previous slide. You're going to get sensory input into the body in one way or another. So maybe that's these neurons, these optic nerves in your eyes that allow you to see things. Then you're going to have integration that takes place in the brain or within the spinal cord, and that's going to be performed by interneurons. And then you're going to have motor outputs performed by motor neurons, which, again, in the case of this picture, just talking about the movement of muscles, but this could mean stimulation of an endocrine gland or all sorts of different things within the body. But you also notice, again, that central nervous system is involved in processing, while peripheral nervous system is involved in the inputs and outputs. This is a cool picture. I like this. This is probably a little more detailed than we would need, and I'm sorry if you hear my dog Samson jingling in the background as he's shaking and doing all kinds of stuff here in the room with me. But I mentioned a moment ago, sometimes the integration, that processing step, can even happen within the spinal cord. It doesn't even require your brain. And that's what a reflex is, right? So, we're all familiar with the quadriceps reflex, which is like when you get, your knee banged by the doctor here. When this happens, what we see is a stretching of this quadriceps muscle right here. Because we've put pressure on this tendon that's running right in this area. So when this gets stretched, your body doesn't like that very much, right? But that sensation of being stretched goes back in here, and here's your spinal cord, this big structure here. And that stretching comes to the spinal cord, and without even going to your brain, your body integrates this and realizes, wow, we don't want that muscle to stretch. If it stretches too far, it could break. So what happens is it sends a motor output back down here through these red tracks to cause the hamstring muscle on the underside of the leg to relax. and the quadriceps muscle here at the top to contract. And that causes you to kick your foot. Right? In other words, it causes this top muscle, the quadriceps, to resist the stretching, and it causes the hamstring muscle to relax and allow it to stretch. And that's why you kick your foot. And notice when you get to the spinal cord, there's sort of two routes. There's one route that requires, here in green, an interneuron, and there's another route that doesn't, and that's why reflexes can be so rapid as well in the body. Now, if we talk about cell structure, your typical neuron has a nucleus, right? Just like any other cell, it also has a number of other organelles and things. But then the cell body is typically sort of an amorphous shape. It doesn't really have a defined shape to it. And then coming off of that cell body is one long path called an axon. And that long extension is sort of your out, right? That's where information is going to travel away from the nervous cell. On the other areas of around the cell body, you typically have multiple branches called dendrites right here. And dendrites are typically how the information comes in to that nervous cell. All right, now what happens is the information, again, comes in through the dendrites, gets sort of processed in the neuron, and the information travels on down the axon, where it then forms a synapse with another cell. Right, now this target cell down here might be a muscle, might be a gland, might be another nervous tissue, another nerve. But in this case, what happens is those neurotransmitters get released from the nerve cell, or from the axon terminus, so we call it the end of the axon, and move from the presynaptic cell to one of these postsynaptic cells. And then the message continues, or you get a response. So hopefully you enjoyed this week's video. I imagine if Mr. Garvey was teaching this to you, he would call these synapses instead of synapses. And so here's a better picture than what I drew. Again, this is a generalized look at this. The actual shape and the kind of layout of a neuron depends on where you find it in the body and what the job of that actual neuron is. But that's something you can, honestly, you don't have to worry about learning until med school, probably, if you want to get into this stuff a lot more deeply. But again, here's our cell body up here. Dendrites are bringing this information in. Then that information can travel down the axon, and then we end up with this synapse we see here, where the signal is moved by neurotransmitters into the next cell. Now, here comes the tricky part. All right, the physiology of the neuron, the way the neuron works, is where things get complicated. Because the question becomes, how does information move from dendrite down axon to the end of the axon and then into this neurotransmitter thing, right? And of course, this is AP biology, so we have to get into more detail, right? Now, the way that this actually works is through ligand-gated ion channels. Whoa, right? We mentioned these, you probably remember building your little model of a membrane on a piece of cardboard for me out of noodles and stuff that I bought at Hobby Lobby, right? And one of those things that you built in your model was a ligand-gated ion channel, which is essentially, right, if we've got our membrane here, it is just a channel embedded in the membrane that requires binding of a ligand for it to open up and allow stuff through. Right? Now, these allow ions through. So something like Na plus or Cl minus or K plus, right? One of these ions. And ions, by nature, have varying concentrations inside and outside the cell. We actually mentioned this way back then too. I think it was like chapter 5 that we were on at the time, that there is actually a difference in the charge based on these ions inside of a cell and outside of a cell. Well, now we have a name for that. And the name we have for that is membrane potential. The idea that there's potential energy, right, in this different kind of charge, plus or minus, across a membrane. Now, most cells in your body are at resting membrane potential, hence the cute, sleeping picture of Charlotte when she was a brand new baby, with her weird, fluffy red hair that she had. And the resting membrane potential for most cells is about -60 to -80 millivolts. Now, again, this is a ridiculously tiny amount of charge, right? Tiny amount of voltage. So it's not like one of your cells can shock you. But -60 to -80, and what this negative indicates is that the cell is slightly negative on the inside, right? And slightly positive on the outside. That's what the negative number means. And so a resting neuron would be a cell that is sitting around this -60 to -80 millivolt potential or charge, and it's a cell that's not sending a signal, right? So it's resting, so it's not active. Now, the way that a nerve cell can send messages is by messing with this resting membrane potential. But first, we got to talk about how you get a resting membrane potential to begin with, and how you can mess with that potential once you have one. Now, as I said a moment ago, resting potential means that the extracellular fluid outside the cell is a little bit more positive, while the inside is a little bit more negative. Now, this is sort of a part of this, as is the next note here. A typical neuron has a high potassium ion concentration inside the cell and a high sodium ion concentration outside the cell. So again, if we're looking at a cell, we're usually going to see more K plus inside and more sodium outside. Now, other ions are also going to be around. As you can see here, chloride is a little bit higher outside the cell, extracellular, but you do not need to memorize this stuff at all. Certainly don't memorize these numbers. The idea that I want you to know is that there is an equilibrium here, right? I probably shouldn't even use the word balance, right? This is an equilibrium, which means it's not equal. But the combination of all these different ions, sodium, potassium, chloride, calcium, in the cell, sets up the resting membrane potential that we see. Now the question becomes, how do you get there, right? How do you get a negative resting membrane potential? And again, just the fact that the membrane potential is not zero tells us that things aren't equally balanced, right? For some reason or another, we've got more negatives inside the cell. So there's a couple reasons that cells are made this way. One, hopefully you recall, so there's a good point of review from first semester, and that's a sodium-potassium pump. We had a picture just like this in our notes. It had more steps to it. It was a bigger illustration. But remember, sodium-potassium pumps are active transporters found on most of the cells in your body. And they transfer 3 sodium ions out of the cell and then return 2 potassiums back in. Now, you guys are smart. You don't need me to tell you this, right? If we take 3 positives out and we only bring 2 positives back in, now we've got a smaller number inside the cell, right? So this has a slightly negative impression on the cell. But this is actually not, even though some people kind of think of it this way, this is not what really sets up the membrane potential on most cells. the bigger influence on membrane potential are just ion channels, right? Pores in the membrane that allow ions to diffuse in or out of the cell. And every ion channel is selectively permeable, right? Another term from first semester. Basically, what that means is each ion channel is specific to a particular ion. So you will have potassium channels, right? And you will have sodium channels, along with chloride and whatever else, right? Now, these channels can be open or closed in their normal position, right? And what we know, that's kind of an important note here, is that a resting neuron will have lots of potassium ion channels open, which allows outflow from the cell. So what we tend to see is a lot of these potassium channels are sort of open all the time. It allows, positives to leak out of the cell. Meanwhile, right, a resting neuron will have very few open sodium channels. So what tends to happen is we block sodium intake, right? This means that we are allowing positives to leak out and not allowing positive back in. And this is what tends to influence resting potential more than sodium potassium pumps that are active and require energy and all of that. It's just sort of a default setting for these ion channels. We allow positives to leave, but we don't allow positives to come back in as easily. And over time, again, what that does is it sets up a slightly negative inside and a slightly positive outside. Now, aren't you glad we didn't try to cover this stuff back when we covered diffusion and osmosis and whatever? That was already heavy enough without laying all this on there too. So, all right, this is what the membrane would look like, right, of a neuron that's at rest. We've got some sodium-potassium pumps here, right? In purple, we've got these potassium channels, which again is K+, right? In orange, and we've got these sodium channels in sort of this cream color or whatever, right? And if you just look at this picture, right, you notice we've got 4 open potassium channels allowing positives to come out, while we only have 1 open sodium channel allowing positives to come back in. Right? And then we got the sodium-potassium pump doing its thing too. Well, this means that we're going to have a lot more positives going out. And we're going to set up this membrane potential. So you see their little positives on the outside of the membrane and their little negatives here on the inside of the membrane. Now, I put this little side note down the bottom corner, right? Eventually, the negative buildup inside will resist the movement of potassium out. This is kind of how equilibrium is established, is eventually, charge attraction will start to play into this too. So the potassium won't want to leave the cell because the positive potassium is sort of attracted to that negative interior of the cell. So the question becomes, right, how does a neuron become activated? Right, how does a neuron transfer a message? There's always some flow of ions across the membrane, but in order to activate a neuron, we need a change in membrane potential. Right? That's how we transmit information or activate a neuron. And for that, we need those gated ion channels. Right? Gated ion channels that were open or closed to allow the rush of ions in or out, kind of at a particular moment, or a sudden rush of an ion in or out of the cell. So in other words, in order to change membrane potential, we have to change the membrane's permeability to ions. So we got a couple terms. Again, just the term polarization by itself. All right, you're kind of familiar with, hopefully from chemistry and from things we've done too, that if something's polarized, that sort of means that it's charged, right? That's not, a lot of scientists would hate that I just said that, but, a polarized membrane means that one side has sort of a different charge than the other. So we've been talking about resting membrane potential, all right? And again, a membrane that is at rest is going to be about negative 60 millivolts. There's a range, but we'll work with the number negative 60 here. So there are a couple different things that could happen, right? The first thing that could happen is we could actually hyperpolarize a membrane. If we hyperpolarize a membrane, that means that we're going to make it more negative. So to hyperpolarize, all right, would mean to make this like negative 200 millivolts or something like that. And the way that you would hyperpolarize is you would cause gated potassium channels to open, allowing more positives to flow out of the cell. And this is basic diffusion, right? The potassium would diffuse out of the cell down its gradient. If this happens, that's more positives going out, right, of the cell, and so the inside of the cell becomes more negative. And that's what hyperpolarization would be. But what we're really interested in is the second opportunity here, depolarization. Depolarization means that the membrane potential becomes less negative. So if we were going to depolarize this, I wanted to do that in blue. So if we were going to depolarize this, it would mean making it something more like 0 millivolts or even positive 50 millivolts or something of that nature. Now, the way to depolarize is opening gated sodium channels, allowing sodium to flow into the cell down its gradient. So in this case, we're going to bring these sodium ions into the cell, which makes the inside more positive. Now we'll come back to that in just a second, right? Any kind of shift in membrane potential is called a graded potential. And if there is a significant depolarization, we call that an action potential. A massive change in the membrane voltage. And an action potential can be very quick and very sudden, but an action potential is the way that a neuron moves information down its axon or down the length of the cell. Now I'm guessing that your head is just spinning at this point, but just stick with me. Okay, just try to stick with me. So here are some pictures of graphs, don't be scared, of what this could mean, right? So again, we would start at resting potential right here, right? And like I said, this is a range, but we'll call it negative 60 millivolts, right? And it's possible that if we were to increase potassium permeability, we could hyperpolarize. We make the membrane potential just a little bit more negative. Right? And notice that this occurs in this little darker area in my graph, which is a stimulus of some kind. So if there's some sort of stimulus that opens up potassium channels, it'll hyperpolarize. Now in the second picture, we again start out at rest, right? But this time our stimulus, for whatever reason, is going to open sodium channels. It's going to increase sodium permeability. Now notice what this is going to do is it's going to cause a slight depolarization. So our line in this graph goes a little bit higher, right? Less negative, more toward 0. Now we'll talk about this more in just a moment, but let's look at this third picture. This third picture is the one that really shows us what a neuron does when it's activated. So this is what an activated neuron would look like graphically. So again, let's kind of break all this down. We've got a beginning at rest. We've got a stimulus that is a significant depolarization, right? It opens lots of sodium channels. And what this does is it allows our membrane potential to raise to a level called the threshold, at which point you get this massive rush of sodium into the cell and this massive shift all the way up to positive voltage. And then it comes crashing back down, as you can see right here. Now, what action potentials will then do is they will move down the axon. So that graph we were just looking at would be like for one particular little location on the membrane of a neuron. And we can get this quick change in membrane potential that can then travel down that neuron almost like information, like it says here, on an electrical wire, a telephone wire. And this, again, is all about gated ion channels. In this case, we're talking about voltage gated ion channels. So a change in voltage causes a channel to open up or to close. So here in bold, I've got kind of the biggest point. If a stimulus at one point on the neuron causes the opening of sodium channels, the depolarization that causes is like knocking over the first in a big row of dominoes, because it then opens up adjacent sodium channels along the axon and allows this message in the form of a depolarization to move along that axon. And this is sort of a positive feedback loop too, right? The current will sort of amplify or will build up, moving or traveling down this nervous cell. Now here's a bigger version of just that same graph we saw before. An action potential, which again is just a massive depolarization, occurs if the stimulus is strong enough to meet our threshold. Threshold for mammals is about negative 55 millivolts. So if we can move the membrane potential just a little bit less negative, we can get to threshold and trigger an action potential. And an action potential is all or none, right? If a stimulus is not strong enough to reach the threshold, so let me draw a picture here in red. If it's not strong enough to reach threshold, then we just kind of draw it back down. If the stimulus is strong enough to reach threshold, bam, the nerve definitely fires. And you can kind of test this on yourself, or you can experience this yourself, right? Think about being able to feel something, right? You ever been sitting somewhere before, and maybe you're outside, you're at some kind of ball game or something, and you notice that the person in front of you has this fly like sitting on his ear and he's not reacting. And you're thinking, how does he not feel that? Well, the way you feel anything is pressure being put on your neurons. And if the pressure is not strong enough to reach threshold and trigger an action potential, then the neuron doesn't fire and you don't feel it, right? It's the same thing like I like to do. I still do this every once in a while. If I'm bored at something, right, I'll start to try to put something on the person in front of me. So like if Dr. Wong was sitting in front of me in the bleachers, I might like try to put my program from mass or whatever on his shoulder or on his head just to see if he notices it. And it's the same idea, right? And so what you can try to do, if you think about it, is you can have somebody close their eyes, or maybe you close your eyes, and ask them to like touch your arm or something. It's theoretically possible that they could be touching your arm, but just doing so lightly that you don't actually feel it yet. And that's because even though they're touching you, it's not putting enough force on the neurons in that area, those sensory neurons, to trigger an action potential. But once there is enough force, then you feel it. Well, some people's are more sensitive than others. So again, you might be able to feel that ant crawling on your leg, or you might not, just depending on kind of how much force it's putting on your leg. Kind of weird to think about. Now, a depolarization, believe it or not, is actually going to open both sodium and potassium channels. The thing is, those sodium channels are going to open a lot more quickly. So this influx of positives into the cell is what's going to happen first. Meanwhile, potassium channels are going to open more slowly, which will allow pluses back out of the cell, just sort of lagging behind the original influx of positives. So this is how the cells sort of recover, right? This is how they go back to resting potential once they have fired. And what's cool is that every neuron has a little period of time called a refractory period. Once it fires, it can't immediately fire again. This is because some of the sodium channels actually kind of stay locked down. Now, this occurs for just milliseconds. So it's not as though you would really be conscious of or notice a refractory period. But it is true that when a nerve fires, there's a little bit of a recovery period where it has to get back to rest before it can then be fired again. It can't really stay active, and that's why, again, that graph of action potential looks like this, instead of like going up and staying up here or something. That doesn't happen. The refractory period, which is sort of like in this time frame right here, right, has to happen, which means that the cell does go back to rest, at least for a couple milliseconds. So here is a really nice picture that I like from the publisher of your textbook, which shows kind of what is going on. So again, starting here in the lower left, and they've got these numbers on here, which I think are great because they tie us back to this really important picture of what an action potential looks like. All right, so at rest, again, sodium channels are closed and potassium channels are closed, at least the gated ones, right? Again, you have some that are always open to help, set up this resting membrane potential, positive and negative, but these gated ones are, are closed. Now then, at step 2, you start to get a depolarization, and that initial depolarization opens a sodium channel, right? Allows a positive charge to move into the cell. Now, as we rise and we hit that threshold, all of these sodium pumps are going to open, these sodium channels are going to open and allow this influx of positives here in the third stage, where we really go north on this graph. And then after that, you have this refractory period, right? You actually have this blockage of sodium channels, and the potassium channels have finally slowly opened up to allow those positives back out. And so it allows us in the graph to drop back down to normal. And actually what happens is, for a short period of time, very short, again, we're talking millisecond here, we typically do see an undershoot, right? We see the membrane potential become a little more negative than normal, and then it levels back out at rest after that. Now, as we said a minute ago, it's kind of like dominoes, right? So basically, everything we've just talked about takes place at one particular location on our membrane, and then it travels down the membrane, right? So the opening here in level 1 actually triggers the opening in this second little area, which triggers the opening in this third little area, and so on, moving this transmission down the axon. And then behind it, you've got these potassium channels opening up to get us back at rest. Now, one thing that's kind of interesting that scientists figured out quite a while ago, a long time ago, is that a neuron by itself is actually way too slow for large animals like us. In fact, if we had to wait for our neurons on their own to send signals to our brain and then send signals back out, it would take a long time. I always, kind of joke around in class, I feel like, and say, if you were just depending on your neurons themselves, you'd put your hand on a stove, you'd wait 30 seconds to a minute, realize you would put your hand on a hot stove and think, oh man, that hurts. But you'd still have your hand there. And then it would be another 30 seconds to a minute before you were able to send a signal down to move your hand off of that burning stove. Fortunately, it doesn't work that way, right? You put your hand on a hot stove and it'll jerk back off of the hot stove before you even realize what's going on. Now, the reason that works is something called myelination, or the presence of a myelin sheath around an axon. It basically is like, I'm drawing a total blank at the moment, insulator, right? An insulation around a wire, right? And so you've got these cells wrapped around a neuron. Now, it depends on where you are, what type of cell we're talking about. In the central nervous system, so in your brain and spinal cord, the cells are called oligodendrocytes. In your peripheral nervous system, which is what they're picturing up here, these cells are called Schwann cells. And they wrap around the axon to form this little insulator. And so what you see in this little picture, or this part of the picture up here, is you have a number of these Schwann cells lined up covering the axon. And in between there, I'm going to make little red dots here. In between each Schwann cell, you can see that the axon is actually exposed, like they're showing you there. And these are called the nodes of Ranvier, which is fun to say, and it's from French there, right? The nodes of Ranvier. Now, the reason that this allows us to increase the speed of neuron function is because essentially the signal can jump from node to node. So what you get is this great picture where this opening and things of ion channels that we just talked about only has to take place at the nodes of Ranvier, not where the insulating Schwann cells are. So basically our message, instead of having to run in a straight line, the message can just hop and it goes way faster. We call this saltatory conduction. So this means that your neurons are faster or more efficient. And because I'm a word root dork, saltatory conduction comes from the Latin saltare, which means to leap. So basically your signal is leaping from node to node, and that's much more efficient or much quicker than if it was trying to do this without the insulating myelin there. So the question becomes, what happens when we get to the end, right? When we get to the end, there's no longer sort of adjacent channels to open. So what happens instead is when we reach the synapse, we're going to convert the signal into a chemical signal. So this is a really cool electron micrograph image here. The tiny little feet, right, are your axon terminals, so the ends of all these axons. And then the big cell here is actually another neuron that's being stimulated. But that might be another neuron, it might be a muscle cell, it might be a gland. And so we have this presynaptic cell that comes in, right, at the end of its axon, and then we've got this postsynaptic cell, which is our target for our message. When the action potential, right, these opening and closing channels, reaches the end of the axon, the last thing it does is it opens voltage-gated calcium channels, and calcium comes in. Now what this does, so here's my axon again, my presynaptic cell. My message has moved down the axon. And it's going to get to this end point, and it's going to allow calcium to come into the cell. Well, what this increasing calcium concentration does is it causes these vesicles that are just kind of floating around in the end of the axon to merge with the membrane and release their neurotransmitters out into this synaptic space. Right? Now, then you've got, again, your target cell here. Your target cell is going to have ligand-gated ion channels. The neurotransmitter is going to bind as the ligand on those channels and cause a change, right? If sodium and potassium channels get opened here, then positives rush in, and we've got ourselves a new action potential, right? We've got ourselves a stimulus here. This might allow us, again, to keep moving our message, might cause hormones to release from this cell, might cause the muscle to contract. If potassium and chloride channels get opened, this will actually cause a hyperpolarization of the target cell. In other words, this will stop the signal or be inhibitory. So maybe your nervous system also needs to tell a muscle to relax or needs to tell a gland to stop secreting a hormone. In that case, it would use this kind of signal and stop things. So here's a better picture than my drawing. Again, got your axon coming down. As we mentioned, calcium would rush into this cell, which causes these vesicles to merge with the membrane, release their neurotransmitters, which then bind with our ligand-gated channels to have some effect on that target cell. Now I'm going to blaze through this really quick because I don't think this is going to be particularly important for the AP exam this year. But you've got more than 100 different neurotransmitters that you can see inside of a mammal. I'm going to highlight a couple that you probably heard of before. And the main one that we're going to highlight is called acetylcholine. Acetylcholine is important, and acetylcholine actually was the subject of, I think, our first bonus practice FRQ of the year a long time ago, which wasn't so much about the nervous system as it just was about sort of general cellular things. But acetylcholine is your major stimulatory neurotransmitter for motor neurons in your muscles. So acetylcholine is what you typically use to cause your muscles to contract. Acetylcholine, surprisingly, is inhibitory on cardiac muscle. So acetylcholine actually causes the heart rate to slow down, right? And then it's also found within the central nervous system. So it is found, acetylcholine, within the brain. The interesting thing is that nicotine will bind acetylcholine receptors in the brain, and that's kind of why nicotine has its drug effects that it does on people. Now, acetylcholine is interesting because there's a lot going on there in a synapse with acetylcholine, and this is what that question was about way back toward the beginning of the year. Again, if we have this neuron that comes in and it releases acetylcholine, right, ACH, into this synapse, we don't want it to just stay there, right? Like, we don't want this acetylcholine to just keep stimulating this target cell. If that were to happen, let's say this is a muscle, your muscles would lock if acetylcholine just kept stimulating the muscle cell. So what you have instead, right, highlighted up here in this note, is you have an enzyme also in this area. Right, so I'm just going to write enzyme in green, which will break down acetylcholine. So basically, as soon as your muscle contracts, you're able to then relax your muscle because of this acetylcholinesterase enzyme. Well, sarin gas is a horrible poison that can be used on people. As you saw, this is a rather old newspaper article. I don't remember if this was maybe in the 80s, which some terrorists attacked Tokyo Underground rail system and killed a number of people with the release of sarin nerve gas, which was developed, I think, in World War II, if I'm not mistaken, as a potential weapon. And what sarin gas does is it blocks acetylcholine esterase. So it comes in and it inhibits this enzyme, which leads to what's called tetanic paralysis. If you don't have this enzyme, your muscles stay locked. And most importantly for your body, the diaphragm locks, and so you can't breathe, and so it can kill you. Another interesting thing to bring up as a sort of real topic is botulism. Right? Botulism is a disease that bacteria generate this toxin, and the toxin will come in and it actually stops, right? So botulinum toxin here in red actually blocks acetylcholine release. So what this does is this causes what's called flaccid paralysis. All right, you cannot move your muscles, and so botulism, again, which is a bacterial disease, it's rare. You can get botulism by eating dirt. Every once in a while, there's a botulism outbreak because of some canned foods or things that weren't formed appropriately. But this too can be fatal because, again, your diaphragm could fail on you. Now, the reason this is really kind of an interesting note is that you've probably all heard of Botox. Botox is the trade name for botulinum toxin, right? This is used for a lot of different applications. It's probably used more in plastic medicine than anything, but it has other uses too. But if you inject a teensy tiny amount of botulinum toxin into muscles, it'll cause them to relax. And so this little picture up here in the corner is an example of what Botox supposedly can do. A lot of people, when Hillary Clinton was running for president, talked about the Botox work she had because she all of a sudden had a less wrinkly face. But as you might guess, you got to be certified to be able to do this. You can't just run around injecting Botox into people, because if you do, you could cause something bad to happen. Now, just these couple really quick. We're not even going to really talk through this, but some of the other major neurotransmitters in humans, glutamate is really important for memory. So that's found in the brain and the central nervous system. GABA is inhibitory in the brain. It allows you to relax, right? So you maybe heard of Valium before. Technically, that medicine's called diazepam. This actually stimulates GABA receptors, and so it allows someone to relax if they have issues with anxiety and those kind of things. Norepinephrine is actually a slightly modified version of epinephrine that actually serves as a neurotransmitter. And then you also maybe have heard of dopamine and serotonin before. Dopamine and serotonin are both found throughout the brain, help with sleep cycles, help with mood management, help with your ability to pay attention, those sort of things. And one thing that's kind of interesting is a lot of the psychedelic drugs, right? Things like LSD, hallucinogens actually bind to dopamine or serotonin receptors. And so if you get those really highly active, you can get some kind of weird stuff going on in the brain. Last thing here is about the human brain. This is not, I don't think, going to show up this year. I could be kind of wrong, but the brain develops really in three pieces. You've got the forebrain, the midbrain, and the hindbrain, and then it develops into these other regions. So I guess it's not a bad thing maybe to throw these words at you, but I really kind of doubt that these will matter much in terms of answering AP questions. The cerebrum is sort of like the big, front part of the brain. You've got these two big hemispheres connected in the middle. This is largest in humans, and so your cerebrum allows you to have really high order thinking, planning, things like that. The cerebellum is found kind of in the rear part of your cranium. This is really important for balance and fine motor skills. Diencephalon is sort of found internally up here. This is your thalamus, hypothalamus, epithalamus. As we saw last time, the hypothalamus is actually involved in some of the neuroendocrine stuff. But this is also really important for maintaining metabolism and body temperature, things like that. And then the brainstem, which is exactly where it sounds like it should be, at the base of the brain, is sort of like your automatic function area. A lot of nerves move through the brainstem, and brainstem damage is almost always lethal because brainstem helps control things like breathing, heartbeat, stuff like that you don't have to think about. Here's a picture, little baby, but all those different pieces, diencephalon, and in the middle, brainstem down here, cerebellum, lower back part of the brain. It's the wrinkliest part, and the cerebrum, which again is not nearly this large in other animals. Cerebrum itself, or the outer part of it, the cerebral cortex, is broken into 4 lobes, which I'm sure if you're in psych, you had to learn these really well. Again, I don't think it matters here now, but you got the frontal lobe in the front, temporal lobe on the sides, parietal lobe kind of more the top of the head, and occipital lobe kind of toward the back. Understanding these areas is good if you go deeper into biology, because then you can start to translate how damage affects people, right? So if you have damage to the lower back part of the head that causes some kind of cerebellar damage, people are going to have trouble with balance or fine motor skills like writing or things like that. Same thing here, occipital lobe has a lot to do with vision. So if a person has a blow to the back of the head, might cause some issues in their ability to see. I just put this in here too. Don't study this. This is just a weird, creepy picture. I always thought these pictures are always found in medical textbooks, that we've really been able to map or chart the cortex. So like this orange part here in the brain, if we take it kind of with the side view, this is the primary motor cortex, the motor neurons. And they've associated all the different parts of the body with different places. So kind of up at the very top toward the middle, this is where you've got like the neurons that are responsible for allowing you to move your knee, right? And coming down the slope a little bit, here's the neurons that allow you to move your hand. And so again, we can track kind of if there's damage in a certain place, what will that look like in that person? And then just behind that there in blue, you've got the major part of the sensory cortex. And so it's a similar kind of map, but it shows you here's where sensation is associated. So if you have some damage to this part of the brain in here, that means that you're probably not going to be able to feel your hand. So I know there was a lot, particularly this last stuff about the brain, I don't think is as important. What I would most expect, if at all, is that the nervous system is brought up in terms of moving ions across the membrane. So that's it. You did it. You made it through all the material. Thanks for hanging with me. Now let's get ready to beat down this test.